The brain is the most mysterious organ in the human body. We know what the brain does, but we don't know how it does it. One of the major components of the brain is the cell called the neuron. Scientists have tried to examine the DNA of the neurons in order to unlock the secrets of the mind. First, we must understand the genes in DNA. The human genome is comprised of 6 billion bases, the nucleotides, packed into 2 sets of 23 chromosomes, one set inherited from each parent. The DNA codes for roughly 27,000 genes. It was generally believed that genes were almost always present in 2 copies in a genome, but recent discoveries have revealed that large segments of DNA vary in copy number. The gene copy number is the number of copies of a particular gene in the genotype of an individual.
Through the examination of neuronal DNA, it has been determined that neurons have a large number of copy number variations, CNVs, which are alterations in the gene copy number of DNA. Due to the CNVs, genes in neuronal DNA have been found to exist in multiple copies, 3 or more, due to gene duplications. In a few rare instances, the genes were missing altogether due to gene deletions. In a study by McConnell et al. About the Mosaic CNVs in human neurons, it was discovered that these somatic CNVs occurred in a significant fraction of neuronal genomes. In the neurons in which they did occur, there was a predominance of deletions relative to duplications. The overall high mutational load that was found in neurons was due to a small subset of neurons with highly aberrant genomes.
The displayed bar chart shows the number of deletions and duplications per chromosome for 3 cell types. According to this figure, more alterations occurred in the neural cells than other cells, and deletions happen much more often. These results show that mosaic CNV is abundant in human neurons. Unfortunately, the mystery of the brain is far from being solved. Although we have evidence of large-scale copy number variation in neural DNA, the effect of these variations on neurological functions remains unknown. These alterations may result in distinct molecular phenotypes that may influence behavior, intelligence, and susceptibility to neurological diseases.
There was a time when to amplify DNA
you had to grow tons and tons of tiny cells.
Ooh, and along came a guy named Dr. Carey Mullis.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primers. Nucleotides and polymerases too.
denaturing, annealing, and extending.